Welcome everyone to the September 7th, 2021 Open Planetary Lunch Talk. I am Chase. I don't have any lunch today, but I have some coffee. Our speaker today, I'm very excited about, is Fabio Crameri. Talking about scientific visualization, specifically the use and misuse of color, which is very dear to me since, as some of you may know, I'm almost entirely colorblind, so many scientific visualizations don't work for me. It's a source of constant struggle. So I really appreciate you being here and for working on this and for smarting us all up on the topic. So thank you, and when you're ready, just take it away.
Thank you, Chase, and also Mark for inviting me to do this. Yeah, that's good to know that some of you are colorblind, and I guess generally we can expect that. So this is partly about this, making sure everyone can read your figures, but also to not distort the data visually. I think those two are the main reasons we started this whole project about how to prevent the misuse of color in science communication. So this is largely based on a paper we published last year in Nature Communications, and I wrote this with my colleagues Grace Shepard and Phil Heron. And most of the information you see here is also available on my webpage. And in general, it's interesting to see the discussion on Twitter as well. So my Twitter handle is there as well. So let's get started. If I manage to change the slide, yes. So this is an outline over my talk. I will start by telling you, and I hope I don't need to convince you, that visualization is important. This is especially true for scientists. It's important that we all try to avoid common visualization pitfalls. And here I talk especially about the ones related to color. And then I will go through the advantage, the availability, and the application of the scientific color maps, which are perceptually uniform and color vision deficiency friendly color maps. And then give a conclusion over what we've gone through, and also an outlook on what is still needed and where we go from here. So let's start. So good science won't be seen without good design. So if we don't make our figures nicely and design them properly, we might lose many readers actually. And also good design and good figures help to memorize what we've seen. So it's a very important part of science. The visual language is also super capable, because it spans not only generations but also cultures, and has basically no barriers. So everyone can see, for example, the heart, the emoji heart, and know what it's meant. And it's a bit the same for figures in general. And then also these days, there are so many papers published and people don't have time to read all of them. So what they usually do is read the title and then scan through the figures first. And if they don't see anything they like, they might not read the paper at all. So making figures is an important aspect of science. And indeed, the images are really the hook that draws the readers in, as the shark can totally see it now. So visualization is important. Visualization is also one of the most widely used scientific methodologies. And at the same time, unfortunately, it's also one of the least widely taught scientific methodologies. So I went through my whole university as a grad student. I never had a class on how to do visualization. So basically I missed out on one of the most widely spread scientific methodologies. Unsurprisingly, I also made some mistakes later on, which I then tried to correct afterwards. So the things you would learn in a visualization class are these, for example. You would learn about the highway billboard test. If you create the graphics, just imagine it on a highway billboard and the people's driving by. would only have around 5 seconds to look at it. So if they don't get the main message of the figure in this short amount of time, they won't get your figure. So if you design it based on this, you make already a much better figure. But then, of course, you need to know your visualization tools to actually create a great figure. So Steve Jobs, for example, was a guy who knew his visualization tools. This is why he showed the Apple market share on a 3D pie chart. And unsurprisingly, he put the share of Apple on the green on the front pie chart piece, which to our eyes looks much bigger than the one in the back. At least he put the numbers on as well, so we can actually see which one is bigger and which one is smaller. But he tried to trick the audience, right, by misleading visualization tools. Another good source to see how visualization tools can be misused is the Fox News. They often show bar plots that are not based on a zero baseline. And then, for example, here shown are border apprehensions over time, and it looks as if they increased a lot. Whereas, in fact, if you would plot it properly with the zero baseline, these numbers are so high that the change is hardly visible and doesn't really matter. And this is more clearly shown here, again on the left-hand side, a bar plot that is not based on a zero baseline versus the proper visualization of this data set. And then you can see there is basically no significant increase in the data, whereas on the left-hand side, it looks like there would be. Another good example from Fox News. So here they just simply skewed the axis on the left-hand side. So at the bottom, there are COVID cases. The change between these ticks is 30, on top it is 50. And so they managed to make the graph look like it was just a simple linear increase and not an exponential one. And they managed to do this by squeezing the axis, which is actually a distortion of the data. Even worse, they made one point there look as if it was going down heavily, whereas indeed this is just due to a wrong or a distorted scale on the left-hand side. So this is the main thing, a main pitfall that cannot be done, but is often done. Figure scales are not here to be played with. So they're not here to highlight data or in some way interpret the data. They're just here to represent your data, and they need to be fixed. Because I imagine you would try to make a graph with an x-axis like this, where the spacing between 0 and 1 is much bigger than the spacing between 1 and 2. You would strongly distort the graph you show, right? And then if you would try to publish this, the first thing would probably be the editor saying, no, you can't publish this like this, please redo the figure. And if not the editor, the reviewers would point it out and so on. The fact is that almost any one of us has already published a scale like this, and I've done it with the rainbow color map or a similar unfit color map. So here is the rainbow color map, and the x-axis at the bottom is the actual representation of this, just translated to a position axis. So you can see the actual distortion, and this leads to very strong artifacts that we introduce on top of our data. To do this analogy, you just need to understand what the color bar actually is, and it's nothing else than an axis. So it replaces a third dimension axis, which we have on the left-hand side. And thanks to the color bar, we can just show a three-dimensional data on a 2D plot, which is very convenient, of course. but we need to make sure to understand what the color bar then stands for. It's the axis, and we cannot change the color gradient between each of the ticks. So this must be constant, as well as the space between the position axis needs to be constant. So this is rainbow. And the distortion that is introduced by these unfit color maps becomes really only clear if we know the data beforehand, if we know how something looks like. So I guess most of you haven't been to space and looked at the Earth, but most of you have probably orbited around the Apple and seen the face of Marie Curie. So you can see that with the rainbow coloring, all of these items are strongly distorted, whereas if you choose a scientific color map, like on the right-hand side, Betlow, then you see the original picture is reflected really well. And there's no visual distortion. And if you need a more, even a more familiar image, found one on the webpage here. I believe this is Nicolas Manot, who is co-founder and president of the Open Planetary. And I, again, colored his face once in the MOLA color map, which is often used for representing Mars topography. And again, you see it's hugely distorted, which is a shame. On the right-hand side, you see Betlow color map, which represents, the face quite flawlessly. And this is important because when I looked for this picture in a higher resolution, I did a Google image search, and it turns out that his face looks similar to some former sexiest man alive, or even Captain America. So we better not distort this face or our data. So color gradients need to be uniform, and they need to be uniform perceptually. This is very important, and this is just a fact, and we cannot play around with that. So to understand how we perceive colors, we need to understand not just our eye, but the whole visual apparatus. And this starts actually with the light source, the colored object, the background of this object, and then our eye, and in the back of our eyes, the cone cells and the optical nerve and the optical cortex. Only if we consider all of these aspects, we can really understand how we perceive color. There's two main aspects we need to keep in mind. So first, there's no uniform color perception among individuals. So we all see color a bit different. And then there's also physiological deviations that can actually shift color perception quite a bit. And this is what we then call often color blindness. So what we need to do to develop perceptually uniform color maps is to quantify basically our human visual perception. And this is kind of complicated. But fortunately, there have been some clever people that already put a lot of work in it, and they derived some color spaces that can easily quantify, or that do attempt to quantify the human visual perception. So we can use these. One is called CCAM02, and they describe all of the colors basically by their lightness. Their red and green and yellow and blue correlate. And from this, we can derive this color difference metric delta E. And this is basically just a measure to quantify the difference between two different colors. So it allows the calculation of how our color map changes in terms of color gradient. This is what we did here in this figure. So you see delta E, the change of color. It should be constant across a flat line across the whole color map, which is almost the case for Betlow, but it is heavily uneven, for example, for Jet, the rainbow color map. And this then introduces a huge error, which can also be quantified. For Betlow, this is kind of insignificant, whereas for chat, this can be up to an error of 8% across the data range shown by the color map. So this is easily your biggest error in any data set, right? So if you imagine you would show a flat gradient, a flat graph with the backlog color map, it would still look flat, of course. But when you would show a flat slope with the chat color map, it would look like hugely bumped, as it's shown here again. So this is important, of course. If you imagine you would display surface topography on a planet and then think that there is a hugely bumped surface, whereas there is just a flat line. Another way to visualize this distortion is by just adjusting the color bar limits, which is done here for the same data set. So this is just one static data set, just shown with the varying color map limit. Now on the left-hand side, and with rainbow, you see that the data is locally highlighted and hidden, depending on where the colors come to lie. Whereas on the right-hand side with the scientific color map, you always see the same data, just with the varying color contrast. So you don't distort data. And then, of course, I guess many of you have done this. If you go on Google and search Mars topography, you see these quite shocking pictures where everything related to Mars is basically shown in this rainbow color map, which is, yeah, as I mentioned before, the molar color map. So here it is again. I replot the data with this particular color map, and you can already see it kind of introduces artificial boundaries. It has some unintuitive coloring. For example, white versus pink versus orange. It's not intuitive for us to say which one is higher and which one is lower value. And this is important because most, or I would say very many figures of Mars topography are shown without the color scale. And then, of course, some people don't like the blue because it kind of indicates that there was water or there is water on Mars. If you compare that to a scientific color map here in La Jolla, for me, it looks more like Mars. Plus, you can also see more of Mars. A general comment about why they use rainbow color map is that you can see more, which is not true at all. You see more artifacts, but not real data. So if you compare these close-ups, you can see that many craters, and especially those low amplitude features, are totally hidden when Mars is displayed in the molar color map. And the same applies to the moon as well, no surprise. Again, some craters are not shown at all, and you can't see these small scale variations in surface topography at all. Whereas with the scientific color map Turku, you can see them. Plus these color maps, the scientific color maps, they are also perceptually ordered, so intuitive, color vision deficiency friendly, and then also readable in black and white print. So these are basically 4 very good reasons to use them. Another example on the Earth, surface topography, bathymetry, and land topography is often shown using a color scale like is shown on the top here, where you have this very skewed part showing the surface topography just above the sea level. And this is a huge exaggeration of surface topography at low altitude, which makes some mountains on the land look like it was much higher than the topography in the sea below the ocean. Well, this might make sense for some applications. Often it does not make sense. And again, you see more detail with the scientific color map, for example, in this spacing here. So a key color map property is perceptual uniformity. So we can't play around with that, and we cannot distort our axis. Another one that Chase would agree on is color vision deficiency friendliness, and then along the same lines, black and white readability. So to go back to this image here, some people see it, so this is deuteranopia simulation. Some people see it like this. And again, you can see you start to have this repeated colors, which make the whole figure basically unreadable for people with this version of color vision deficiency. The same for protanopia, and then also for tritanopia. And then in black and white, of course, everything basically is smeared out and looks the same in this region here, and has some strong gradients on the left hand side and the right hand side. So using the molar color map here, it's not readable at all, whereas you use the scientific Bedlow color map, you can still see the original pictures. Of course, it's not just my scientific color maps, which is Bedlow part of it, but also perceptually uniform color maps by other people, like Viridis is a well-known one, or Cividis. They're all color vision deficiency friendly, whereas if you compare it to Chet, it is not. And then another key color map property is intuitive color order. So you want, if you just look at a figure without looking at the scale, you want to see or understand which values are higher and which values are lower, and how they are connected to each other. So you can do this with the scientific color map at the bottom, and both via the color and the lightness, you can order them. So if you just take a couple of individual colors out of this color map, you can easily order them, right? When you try this with the Chet rainbow color map, you can neither do it with color or lightness. So if you try to do that, you most likely fail. So to summarize, the scientific Bedlow color map is perceptually uniform, rainbow is not. The Bedlow color map is perceptually ordered, rainbow is not. The Bedlow color map is color vision deficiency friendly, rainbow is not. And Bedlow color map is readable in black and white, whereas rainbow is not. So I guess the conclusion is Bedlow is scientific and rainbow is not. So we can easily scrape it and not use it anymore. Because all we want to do is basically communicate the data without distortion, as intuitively as possible, and to as many readers as possible. I think we all agree on that. There's one more key color map property, which is availability. And again, I guess you all agree that it should be openly available. So here's the scientific color maps. It's the whole suite. It's now version 7. And they are all perceptually uniform, ordered, and color vision deficiency friendly. And on top of that, it's a complete suite. It has all necessary types and classes, and they are versioned and citable. So if there are some problems, I can easily update them. And as I said, they are freely available. So those are the scientific color maps. As you can see, I tried to cover all the different types, etc., and have also kind of broad spectrum of colors, so you can fit them to whatever you want to show. So how do you use the scientific color maps with your data? So these are the different classes again. So there's sequential, diverging, multi-sequential, and cyclic. And let's just go through quickly what they mean. So the sequential one is basically just showing a data field that goes from low values to high values. As for example, here, I think that's the depth of some earthquakes or something. The diverging class is used to represent zero-centered data, where you basically show, for example, a temperature anomaly, as is done here, where you show the cold anomalies and the warm anomalies on the same figure. So you basically show these two kind of connected data parts. And then there's the multi-sequential one. They are very specific, for example, to surface elevation, where you again have two kind of separate parts. Here it's ocean bathymetry versus the land topography. And you just show them with the zero-centered color map again. And then the cyclic class, it's used for circular data, for example, angles on the surface, orientations, or INSAR measurements, as is done here. So this is the continuous type, and then to complicate things, there's other types as well. So there's discrete type, for example, where you just pick 9 or 10 different colors with the same color difference between them across the continuous color map. And this is useful because sometimes you want to kind of be able to pick what value a certain range in the data has specifically, which is sometimes hard with the continuous color map. So you use a discrete type. And then there's also a categorical scientific color map type. For example, Betlow S, as is shown here. And this is useful to just plot lines or points in different unordered colors. So I created this categorical color map, which has up to 100 different colors for these lines. And of course, the more lines you need, the closer these colors get to each other. But they have a maximum color contrast depending on how many lines you would show. And again, they are color vision deficiency friendly, readable in black and white as well. So these are the different color map types and classes. Now, if we want to know when to use which, you can also use this user guide that we provided alongside our paper in Nature Communications, or you can download it directly on my webpage. It gives you a kind of flow chart for any given data set. You just go through and then you see what color map class and type you can use. And then also, if you go further down, you even see some information on how to choose the best color combinations, whether you should have a dark part on the left-hand side with the lower values or on the right-hand side, and so on. Because, yeah, background color is also important. We perceive foreground color differently depending on the background, as you can see here. So the font for both these words is the same, whereas it looks, at least to me, kind of different. So background appears brighter because it has a darker background, and color appears darker. And the same is true for color itself, for hue. For example, background looks as if it were yellow and color looks as if it were green, but it's actually the same color. So this is another thing that should be kept in mind. And there's a kind of mind-blowing video, which is in the supplementary of our paper again, which you can look up. So yeah, you can use these guidelines to effectively apply color maps. But then how do you use the scientific color maps with your software? And again, here's the entire suite, and I provide all of these color maps in different formats, actually more than 20 different formats, which is kind of ridiculous, but all the software apparently use different formats for their color maps. So I made sure you can import all these color maps into different programs listed here. And there are many tools as well that are extremely convenient, for example, for Python or Julia, that you can just use and basically provide you the scientific color map as if they were building. And then there is also more and more software tools that have actually built in the scientific color maps, which are listed here. And of course, this wouldn't be possible without all the help of this amazing community. For example, Mark helped a lot with implementing the scientific color map format for Python, and there are so many others that helped with that as well. And then, yeah, we just need to keep pushing, I guess, to spread the word. And for this, again, we need, or I need your help as well. So everyone who uses it helps a lot. So be like Captain America and lead by example. Use the scientific color maps. Say no if people use rainbow, for example, in peer review or even to your peers and colleagues. I have provided on my webpage a frequently asked question site with all the answers to the most often made statements by scientific color map critics. And then we also made this convenient coffee room poster that you can hang up to remind yourself and others about the importance of using scientific color maps. And I don't know if you can already go back to coffee rooms, but once you can, maybe you want to do that. I haven't told you the real solution to the whole problem of the misuse of color. I think it would be to just teach visualization, which is, as I said before, a fundamental scientific method already at university level or before. I think this is really key, and otherwise we will keep falling back into bad habits and people won't keep using scientific color maps. So to recap, visualization is a fundamental scientific method, and it's very important that we learn about it or get people who know about it to help us. Color perception varies not just between hues, but also between people, which we need to, remind ourselves. A color map is the fundamental property of a color axis. So always, if you think about color map, think about the analogy to a position axis, like an x-axis or a y-axis. And then it becomes also clear that you cannot fiddle around with the color map because it's the property of the color axis. To interpret or highlight parts of your data, don't use the color map, use other visualization tools like superposed contour lines or graphic indicators like errors or squares or something. And then finally, to excel your research, make sure that you display your data accurately and inclusively. So thank you very much for your attention, and happy to answer some questions.